So if you still remember that biopharmaceutics examine the interrelationship of physical chemical property of the drug or dosage form, okay, onto the rate and extent of systemic drug absorption. In other words, on bioavailability. Okay. So in biopharmaceutics, besides understanding the bioavailability concept, we need to understand as well the physical chemical property, which involve the manufacturing process, okay, and also raw. Okay, so when you talk about pharmaceutical manufacturing industry, they are highly regulated, which they need to have the controlled GMP condition, and they have extensive quality control and quality assurance testing with tight and stringent limits to ensure safety and efficacy of the drug to the end user. So sample list of testing for QC will be including raw materials as well as finished products. So in raw material, they will test the following items with identification of the active, the related substance of the excipient, for example, is there any impurity in the active, any solvent, moisture, purity of the drugs, heavy metal, and microbial limits. Whereas on finished products, they need to identify as well the active ingredient, the drug content, the amount of the active ingredient, content uniformity, related substance, for example, the excipient, the same thing, microbial limit, stability of the finished product, so they need to subject the product into stress test, okay, hardness or friability. So other additional tests for the tablets and capsule include disintegration and dissolution. Okay, for an immediate release dosage form, they need to disintegrate within 15 minutes. Okay, whereas they need to release the drug more than 85% in 30 minutes. Okay, this is the requirement for immediate release dosage form. So with that, for immediate release products, for example, there are possible differences. For example, we have a drug from a branded company, okay, let's say from Pfizer, and this is the drug that you have developed in your own generic company, okay. You use the drug particles, excipient, the same manufacturing process, you use the same equipment for tableting, and maybe your site of manufacturing is different, so your batch size is different. Alright, okay, so if both tablets behave, okay, therapeutically same, you will say it is therapeutically equivalent. Okay, however, this is not always happen when a drug is compared to a branded medicine. Okay, so for example, when you subject to similar regulatory control, means you also have the same QA, you have the same quality control, the same condition requirement for the GMP industrial environment. Okay, however, your drug may be performed differently, right? So when we compare the price for a generic company, of course, and a branded company, or we call it the branded as innovator. Okay, generic and innovator company. Okay, so for clopidogrel, okay, from Plavix, this is clopidogrel. From Sanofi, the original innovator, is 6 ringgit 80 cents per tablet, and you compare with Ranbaxy, a generic company. Okay, they take only 2.5 rm per tablet, which is much cheaper. Okay, and same happened to atorvastatin and simvastatin. So by using a generic drug, you may save 60 to 65% of the cost of therapeutic. Okay, so when we compare generic and the patented drugs, so generic drug and patented from the innovator or the branded one, okay, they have the same active ingredient, the same dose, and the same dosage form, for example capsule, tablet, etc. Alright, however, they can still have differ in efficacy, especially for solid dosage form. Okay, this is all due to the differences in bioavailability. So when you recall back the factor affecting bioavailability, then you will understand why a solid dosage form can have differences in bioavailability, even though they have the same drug, same dose and dosage form. So here comes bioequivalence study. So here we are trying to study, okay, or compare, okay, the bioavailability of innovator drug versus generic. Okay, and if after we have compared both of them and they are equivalent therapeutically, then it is bioequivalent. Okay, so how do we decide on a generic, whether you want to advise your patient to take a generic medicine? So the best proof for that is a safe and effective substitute for an innovator product, which is from bioequivalent study. So the two products consider bioequivalent if they have the comparable rate and extent of drug absorption, which is they have the comparable bioavailability. So there are a few methods that we can use to assess bioequivalent, okay, which is based on measuring the drug concentration in plasma or serum, or based on pharmacodynamic study, which are measuring the pharmacological response, or based on clinical trial, measuring the therapeutic response, or by using in vitro study. And in the subsequent slides, I will focus on the assessment of bioavailability by measuring the drug concentration in plasma or serum for bioequivalent. conduct a BE study, BE is bioequivalent, you need 5 main events, okay, which is you need healthy human volunteers, around 16 to 30 subjects, two-way crossover study, I will explain that in the next slide, and you need to test the blood sample over 24 to 3 days, 24 hours to 3 days. Then the sample is taken for analysis for the drug concentration, and eventually pharmacokinetic analysis. So for human volunteer, it should be healthy. Both gender can be included, okay, but male is preferred when you are collecting urine sample, okay, and they should be between 18 to 55 years old, BMI of 18 to 30, and the minimum of volunteer should be 12 and not less than that. Okay, so what do you mean by two-way crossover study design? Okay, so two-way crossover study design meaning that, let's say you have two groups of subjects, okay, let's say group 1, 6 of them, group 2, 6 of them, okay. So in phase 1, okay, let's say in day 1, you ask group 1 to take the generic products, okay, let's say 1 tablet, okay. Then for the group 2, okay, take the reference products, okay, then their blood plasma is collected and analyzed for the active drug for continuously 24 to 72 hours. All right, then they should have a what you call washout period. All right, so let's say washout period of let's say 2 weeks, okay, so all the drugs in the body is completely eliminated. Then the subject enter into phase 2, let's say in day 15, okay, and the same thing, group 1 now take the reference and group 2 take the generic. So each of them become the control of themselves. All right, so after that, the serial blood sample are collected over 24 to 48 hours. Means once the subject enroll into the study, okay, on day 1 to day 2 or day 3, they should stay in the center for blood withdrawal, okay. Then the blood is taken for analysis by using HPLC. This is the machine for HPLC, okay. And the analysis of the plasma drug level, which is in under the HPLC machine, is the one that is very challenging, okay. It is the most difficult part of the study when you are dealing with nanogram or even picogram of drugs concentration in the blood, okay. So usually we need a high-end HPLC system with the UV fluorescent or mass spectroscopy detection, okay. So in the BE, a bioequivalence study center, usually is about 1,000 plasma that they analyze in one study, okay. And before that, so this is the blood sample. If you look at here, this is the blood sample that's withdrawn from the patient. Okay, this is the blood sample, and the drug need to be extracted, maybe by a solvent, before they can be injected for analysis. Okay. So, however, before we start to analyze the plasma concentration, the development of HPLC method, okay, needs validation. The criteria for validation will be accuracy, how accurate the methods, precision, linearity of the detector response, detection limit of quantification, and recovery of the method. Okay, so I assume that you learn about HPLC in chemistry subject, so I won't repeat HPLC in detail. So you may want to revise some of your notes from the chemistry lecturer. Okay, then we should after we got the plasma concentration, then we can plot, okay, the plasma concentration against the time for all the subjects. So this is mean plasma concentration of ketoconazole versus time profile of Nezora and generic. Nezora is the innovator and generic products, okay, with a sample of 12 person. Okay, so in pharmacokinetic analysis, what we are looking for, we are looking at the area under the curve, which is area under the curve here. We also look at the Cmax, okay, concentration here, Cmax, Tmax, okay, time when they achieve maximum concentration, Tmax here, okay, Ke, the elimination constant, which can be calculated. So important pharmacokinetic parameter, which is AUC, area under the concentration time curve, it is a measure of extent of bioavailability. So when we talk about Cmax, it is related to the rate and extent. And talking about Tmax, it is referred to the rate. Okay, so when we have the rate and extent, okay, so AUC focusing on the extent, Cmax, rate and extent, and Tmax will be a measure of the rate. Okay, so what are the bioequivalent criteria? Okay, so means after you have carried out the study with the plasma concentration of a certain AUC and certain Cmax. Okay, so what are the AUC and what are the range of Cmax for an acceptable generic products? Okay, so in this case we compare, let's say we compare the curve for the innovator, this one, and then the curve for generic. Okay, so usual statistic analysis is not suitable to compare the two curves, and usually in this case we are using confidence interval. So we are looking at 90% confidence interval. So for confidence interval for AUC, it should be between 0.8 to 1.25. Okay, so meaning that we are 90% confident that, okay, taking this generic medicine will achieve 0.8 to 1.25 of the mean AUC. Okay, same thing happened to Cmax. If we need to have the Cmax range between 0.8 to 1.25, meaning that we are 90% confident that the generic will produce the Cmax between the value of 0.8 to 1.25 of the innovator. Okay, so have you ever think about why is it 0.8 to 1.25? Okay, it is because the FDA decided that the differences in systemic drug exposure up to 20% are not significantly different. Okay, and if they are not significantly different, why is it not 0.8 and 1.2 but 1.25? Okay, because pharmacokinetic parameter, actually they are not normally distributed. They are normally distributed only when they are in the log value. So meaning that when you transform the exposure parameter, okay, AUC and Cmax, okay, the non-normal curve will turn to a normal distribution. All right, so referring to the normal distribution of log curve, then we take 20% as the limit that is not clinically significant. So it should be 80% to 120% in the log value. So when taking the log value and you anti-log the 80% and 120, it becomes 0.8 to 1.25. Okay, so normal distribution are generally required for specific statistical tests. So that's why the symmetrical 20% has to be in the log transform space, so that the statistical test of bioequivalence will be valid. So referring to this table, please. So when we have, this is our test, okay, and this is the reference, means this is the one that in innovator, okay, and this is the one for generic. Okay, we take the ratio of 2 and then we got this. Okay, this is a ratio. Okay, and this ratio divided by the reference, then you get the percentage. All right, so when you get the percentage, when you log it, okay, it will become normally distributed. Then only you take the 20% as the limit for the log distribution curve. So meaning that here, so we take 20% here, 20% here. Okay, so anything fall between this point A and point B, then they are what you call the criteria is met. Okay, so when you look at here, 80% when you log it, it becomes negative 0.223, and when you have 125, it is also transformed into 0.223, which is normally distributed. That's why we will take 0.8 to 1.25 as the criteria for bioequivalent. Okay, so for example, when you have the generic, okay, the black one, and the Nizora, the circle, okay, and with their 90% confidence interval of 0.96 to 1.23, so this one pass the criteria, and the Cmax is 1.01 to 1.25. Yes, they also pass the criteria because they are within 0.8 to 1.25, within this range. All right, so other examples such as this one, let's say you are given 90% confidence interval of 0.74 to 1.06, 0.78 to 1.06, because this one go lower than 0.8, so it is not bioequivalent. bioequivalent. Okay, so when we have example 3, this is within 0.8 to 1.25, same happened to CMAX, so it is consider bioequivalent. Right? So, however, bioequivalent study meaning that you need to have your generic, okay, between 0.8 to 1.25. So what if the drug being absorbed extensively in the patient, okay, so they will have the higher bioavailability, which is exceeding 125%. So even though you have better absorption, you will still fail the bioequivalent study. So as a generic company, you can't market your drug because you fail the BE study. So for a BE study, so a generic cannot be inferior, but they cannot be better. However, generic can perform actually better than innovator. So this is one of the example of generic of glimicron and dimicron, the innovator, and glimicron, the generic company. And even though the generic company achieve a better bioavailability performance, okay, however, they fail bioequivalent. So if I ask, can a generic formulation perform better than the innovator? The answer is yes, but they fail bioequivalent, okay, because they achieve the much higher absorption. All right. So in this case, even though the generic has performed their bioavailability, which is much better than the innovator, however, they couldn't get their drug marketed simply because they didn't fulfill the requirement for bioequivalent study. Okay, this is another example for as well as the glycoside and glimide. Okay, this is a modified release. Okay, from the innovator products. So even though the innovator has modified the release, but their release is still not better than the local formulated glycoside, right? So with that, we may want to argue why we need to do bioequivalent if our product cannot perform better than a innovator. Okay. Bioequivalent can also apply to nutraceutical, means supplements. Okay, and it is allowed to be better, okay, molecule where bioavailability is an issue, so that we can always improve the formulation to increase the bioavailability, okay, so that the consumer can get their money worth the products. Okay, example of nutraceutical that you can use for bioequivalent study will be vitamins, coenzyme Q10, DHA, EPA. So with that, a company can actually design a special delivery system so to increase the bioavailability of this nutraceutical, okay, nutraceutical, and compare with innovator. With that, they will save their time in preclinical study involving human volunteer, which is having the disease. Okay, this is one of the example that the bioequivalence study that has been applied to nutraceutical, tocotrienol, vitamin E. Okay, and this is the conventional preparation, and this is the generated formulation X. Okay, generic. All right, so this is the generic. Okay, and we found out that the formulation X, which is in generic, perform better bioavailability, higher bioavailability than the conventional preparations. Okay, so with that we may actually reduce the dose. Okay, looking at the serum drug concentration of the new products. Okay, so this is another example that bioequivalence study applied to Nutraceutical, which is coenzyme Q10. Okay, so we compare our product from whole wheat compared to the other two different products, and we found out that whole wheat actually generated a much better product performance of coenzyme Q10. Okay, so based on our experience, okay, immediate release product is easier to achieve bioequivalent, especially when the active have a high solubility, whereas when you have a modified release drug or dosage form, okay, we need to have additional consideration, usually the excipient, and they are more difficult to achieve bioequivalent because of the stringent evaluation. Okay, so go back to biopharmaceutical classification system, which is a scientific framework to classify drug substance, okay, based on solubility and intestinal permeability. Okay, so permeability is drug ability to cross biological membrane. Okay, they should have satisfactory lipid solubility and non-substrate of efflux pump like PGP, so that they can have high permeability. So in BCS classification, the drugs is divided into 4 classes. First class is with high solubility, high permeability, and second class low solubility, low permeability, high permeability. Third class will be high solubility, low permeability. So BCS can be applied to examine a generic product from bioequivalence study. How? So because if you look back, why we need to conduct a bioequivalence study? Simply because when you are when there's innovator and you are the generic, since you have the same active ingredient, okay, same dosage and dosage form, so what are the difference? The difference will be in excipient, and how excipient can affect the bioavailability is physical, chemical properties of this excipient. So when you have the active ingredient, okay, which having a high solubility and high permeability, which their performance is not easily influenced by the excipient, okay, then the drug, the certain drugs, okay, can be exempted from bioequivalent study. So I give you an example. For example, paracetamol is classified as a BCS class 1, where they are highly soluble and highly permeable. Okay, so even though we formulate them in different manufacturing sites, they are likely to have the same bioavailability. Okay, with that, we no need to perform bioequivalence study which involve human volunteers. Okay, so in this case, they can ask for bio-waiver. When they need to ask for bio-waiver, they need to meet the 3 criteria, which is the solubility, permeability, and in vitro dissolution test. Okay, with that, they need to compile documentation and apply for the bio-waivers. Okay, so scientific basis for bio-waiver: BCS class 2 drugs product with rapid in vivo dissolution relative to gastric emptying. So the rate and extent of bioavailability is unlikely depend on dissolution or GI transit time, but it is depend on the gastric emptying. Okay, so as long as excipients do not affect the absorption, the drug should perform similar as the innovator. However, we need documentation to support these bio-waivers. Okay, so when dissolution is very rapid, okay, this is very rapid, so the absorption is mainly influenced by gastric emptying. Okay, so essentially, to compare the drug absorption of two products where dissolution is rate limiting step, okay, means bioequivalent is essentially to compare the product whereby the drug is rate limiting, the dissolution is the rate limiting step. So according to BCS classification, which class where dissolution is rate limiting step? So usually those drug with poor solubilities, which is class 2 and class 4. Okay, so however, for class 1 drugs, as rapid dissolving dosage form, drug absorption is limited by only the gastric emptying, which is dependent on individual, not dependent on the manufacturing site. That's why in this case, bioequivalent can be waived. Okay, under such circumstances, bioequivalent evaluation is redundant, since any difference in between the two formulations is due to the difference in the rate of gastric emptying and not the difference in property of the true products. Okay, so FDA defined that method for determining solubility, permeability, and in vitro dissolution, so that when a company need to apply for waivers, they need to provide document for solubility, permeability, and also the dissolution testing with the method that has been defined by FDA. Okay, so boundary for class 1 drug, which is high solubility and high permeability and rapid dissolving products. So there is a boundary. So how we can consider a drug as high solubility or high permeability? So the drug considered high permeability when we have the fraction of those absorbed more than 90%, means they are highly permeable. So this is to determine using human subject when we compare with the IV dose, because when we want to compare bioavailability, we need to compare the one absorbed with IV dose. Okay, or you can actually use non-human system capable of predicting the absorption, for example using cell line. Okay, when a drug is considered highly soluble, the highest dose strength is soluble in 250 ml or less, medium over the pH range of 1 to 7.5, then it is highly soluble, which you can apply for bio-waiver in bioequivalent studies. Okay, so how a drug can be considered to have a rapid dissolving behavior when not less than 80% of the labeled dose dissolve within 30 minutes. Means within 30 minutes need to have more than 85% of drug release, and they must use 100 rpm and apparatus 2 for 50 rpm. Apparatus 2 is pedal and apparatus 1 is basket. Okay, and the guideline for the medium will be 900 ml of the medium, either using stimulated gastric fluid or HCl without enzyme, or they can use phosphate buffer depending on which is the absorption site of the drugs, or they can use what you call simulated intestinal fluid as well without enzyme. So why is it 900 ml of medium? So because all the from stomach up to the intestine, the liquid in your body for oral drug absorption is 900 ml. So it is fixed for this solution medium to be in the volume of 900 ml. Okay, so any additional consideration for bio-waiver will be, it should be the excipient do not affect the absorption, and they should use USP approved excipient and also use at the normal amounts intended for functions. So abnormally large amount may pose some problems, so they will not get any the application of the bio-waiver will be failed. Okay, however, BCS class, okay, for bio-waiver waivers is not applicable to drug with narrow therapeutic index, okay, or they are non-GI tract gastrointestinal tract absorption. For example, when they are intended for buccal delivery. So this means that we cannot utilize the BCS classification to apply for bio-waiver if the drug is a narrow therapeutic drugs or they are intended for non-GI tract absorption. So bio-waiver also applicable to class 3 drugs. So BCS class 3 drugs, product formulated with rapid in vivo dissolution related to gastric emptying, because the rate and extent of bioavailability is unlikely to be dependent on distribution and dissolution, but they are related to permeation rate, which is inherently to the drug molecule. So in this case, once again, the manufacturing site, okay, the formulation variable, is not going to influence bioavailability. That's why they can also apply for bio. So if you look at this slide, when we talk about class 3, when this solution is very rapid, because in class 3 you have the solubility is very high and permeability low, right? So when we talk about class 3, when you have the high solubility, okay, so in this case, this integration, this solution is not a problem. Okay, so in this case, permeation rate is the limiting step. Okay, so That's why when K1 more than K2, permeation rate is the limiting step. That's why a company, if they are formulating a class 3 drugs with generic products, they can apply for bio-waiver. Okay, so to summarize, so justification for bio-waiver will be drug classified as class 1 and class 3 according to BCS. So they are well formulated to achieve rapid dissolution, okay, according to guidelines. So we need a proof for this one. So they need to run in vitro dissolution test. They use suitable excipient which has been approved by USP with the amount of usage, and they are non-therapeutic index drug. Okay, so they are non-therapeutic index drug, and as well as they are not intended for route other than oral. Okay, so you have 1, 2, 3, 4, 5. So it means when they are for buccal delivery or any other delivery, then they cannot ask for bio-waiver. Cannot ask for, right? Okay, so the list of drugs that requiring bioequivalence study in Malaysia includes the first list in Malaysia is nifedipine, cyclosporine, captopril. Okay, second list already in 2000, and those are the drugs. The third list in 2001, theophylline, propranolol, cimetidine, carbamazepine. Okay, then the drugs in 2002, 2004. You don't need to remember all the drugs in specific years. Okay, and the sixth list in December is included here, and we have more to come. So in the total until 2009, 113 drugs are on the bioequivalent list. Okay, and where are the centers of bioequivalence study in Malaysia? So we have centers in USM as well as in UM and Infokinetic company in Penang. Okay, and in USM we have 3 teams, 2 from School of Pharmaceutical Sciences and 1 from the Drug Research Center. As well, you no need to memorize the centers in Malaysia. Okay, so summary, a generic and branded can differ in efficacy due to the differences in bioavailability, arising usually from the formulation and manufacturing variable. So only the two, formulation and manufacturing variable. So bioequivalence testing provide the best proof of switchability and interchangeability of the drugs for a certain disease. So generic can be better but not permitted when they are better. So when you formulate a drug with a better performance of generic, perhaps you need to go through the other pathway for marketing your drug rather than bioequivalence studies. Okay, with that, thank you.